All right, welcome back to the MBP podcast. I'm Calder, and today we're joined by Dr. Benjamin Locke from UHN Research. He's a clinician scientist studying patient outcomes in lung cancer. So, Dr. Locke, thanks for coming on the podcast.
Calder, to you and the medical biophysics team. It's a great pleasure to be here, and thanks for the invitation. I'm excited to chat with you and just, get to know each other.
Awesome. So, can you tell us a little bit about your background and how you ended up where you are in your clinician scientist role?
Sure, happy to. I'd like to say I'm a proud immigrant from New York, across the border up north. So that was a great adventure for my family and I moving here. Before that, I was actually, I'll go back a little bit further. So I went from business school and undergraduate at NYU, New York University, Stern School of Business, then decided to do medicine instead of business, so applied to medical school. So I went to NYU again, the medical school there. Now it's Langone Medical Center in the School of Medicine. And that was where going through actually the first 2 years of medical school is very much like what you are going through, Calder, in your courses in graduate school. Take like biochemistry, pathophysiology, of course, more steered a little bit more towards like medical type of graduate courses, but still very much similar. There are actually some basic science, life sciences graduate students in some of our courses. So that's when I actually got a greater appreciation for how much um, science and discovery informs what we do in clinical medicine. I didn't as fully appreciate that in undergraduate because we just take our general chemistry and organic chemistry. It wasn't yet linked to as directly to human disease. So that was eye-opening for me, just those first couple of years. So that really actually then planted the seed of, wow, that's how these um, medical advances came into being. It was actually all this hard work in the laboratory and understanding the mechanisms of disease. And that really got me thinking about, wow, this is something I haven't yet been able to dive as deeply into with my own hands, other than just reading something in a book. So that really led me towards looking at different um, experiences to enrich my medical training. And along the way, I developed an interest in oncology from personal reasons. I lost a good friend to cancer. I have family members that are cancer survivors. So just the cancer and oncology was a both personal interest in addition to guiding my professional interest in development. So within cancer, like when I was in medical school, um, Gleevec had just come out, and imatinib, so the
Okay.
first targeted therapy. So we learned about BCR-ABL translocation, so I was like, wow, like this is really impacting oncology. I mean, it was before, but this was something that was just quite uh, interesting and it was just happening as I was going to medical school. So I wanted to get involved more on the lab side. So that's when I took a research sabbatical after my third year of medical school. So medical training is 4 years, like 2 more coursework years, the last 2 for more clerkship or more clinical years. Um, so after my third year, I just was advised by some fellow students that were graduating. They were both MD PhDs, so they actually knew basic science and that type of work and could advise me on how to try to get connected or what to, which are labs that would make sense for what I was interested in. So I just cold emailed a bunch of people up the street at Memorial Sloan Kettering. I think it was like 7 to 10 lab heads. So I cold emailed. Only one I think got back to me, and that one was Simon Powell, the chair of radiation oncology, a physician scientist who studies DNA repair and specifically
Mhm.
homologous recombination within the BRCA pathway, the BRCA pathway. So that, you know, make a long story short, that's where I ended up in his lab. Really enjoyed that discovery process. Initially, it was just a year, but then, quite quickly into it, decided to extend it for a second year. So I did 2 years in his lab and then went back to fourth year even continued part-time laboratory work because we had to get my paper out. So my first
Yeah.
basic science paper was published in your fourth year of medical school, and then went on to Memorial Sloan Kettering in my clinical residency with radiation oncology. And that's a 5-year program. The first year is an intern year, so you could do anything you want: surgery, medicine, internal medicine, or like a transitional year. I decided to do internal medicine year. So I stuck around at NYU, and then the next 4 years was up at Sloan Kettering. And then there, because Simon is a huge proponent of just really advancing biology and having us think deeply about that to enrich what we do in our clinical training and practice. So one of the things he instituted when he first chaired that department was to set aside a whole research year during your residency, protect
Cool.
research time of at least 12 months. So I took advantage of that. I hopped back into a different lab, and I wanted to make a difference or understand what makes lung cancer so difficult, so challenging. So I spent that lab research year with Charlie Rudin, another physician scientist. He was chief of the thoracic medical oncology service and still is. And that's where I started merging things from Simon's lab and Charlie's lab into something of my own. So the mission of my laboratory is to improve the outcomes of lung cancer patients, meaning helping them live longer, live better. And we do that through a couple of basic research angles and translational research avenues as well. One is novel therapeutic development, designing the treatments of tomorrow. And in particular, combination treatments we're quite interested in, and those related to DNA repair. So with radiation, with different DNA repair inhibitors, even our traditional chemotherapy or DNA damaging agents. So that really girds a lot of the cancer therapies that we have today, and how do we make them better and more effective? That's one avenue. The other research avenue is mechanisms of resistance. We just see, for many lung cancers, they are treatable initially. We can make patients live 3, 4, 5 times longer with chemotherapy, with radiation, and now with immunotherapy. But for many of those patients, the cancer comes back even after those initial treatments shrunk a lot of the tumors, put them in remission for a while. But when cancer returns, it generally is, or usually is, more resistant to the initial therapies that were given. So what are the mechanisms driving that? And through understanding that, can we either prevent that from happening, or can we now still render effective treatment even in a recurrent, relapsed setting where the cancer has come back, but can we make treatments that are effective against that resistant state? So a bit of a long-winded answer here, Calder. I don't know if I got to the
That was great.
gist
I mean,
of it.
a lot to unpack there. I guess first, I should mention I'm also from U.S., from California, so fellow cross-border immigrant, as
Welcome.
you call it.
Excellent.
Yeah. A couple things. I know you mentioned that there's a sort of a crosstalk between the biological research and the clinical research. So I was wondering, sort of, how does that work? How does your basic science research influence your clinical practice, and vice versa? How does your clinical practice impact your basic science research? Like, what's the interplay there?
Great question, Calder. So how does my laboratory research interact with my clinical role and my clinical practice? I think I'm quite fortunate that I'm able to connect the two in a very tangible way. Not to say this is the only way to quote unquote be a clinician scientist, definitely not.
Mm-hmm.
But for me, I do feel fortunate because it helps me take findings from the lab and actually have a direct route and vehicle to apply it into the clinic. So designing clinical trials, for example, off of the work in our laboratory. That's something we've done recently, looking at a drug known as a PARP inhibitor. We've shown that in laboratory models that it radiosensitizes, it further enhances the radiation anti-cancer effects on small cell lung cancer. So that's led to us testing that concept out in patients. So I'm running that trial here at Princess Margaret. That's really nice to be able to see that directly and personally. and it's with patients I get to treat as well. So that really helps motivate the research in my laboratory, helps inform. We see the clinical challenges, and we can bring those challenges, and this resistance mechanisms. We see this all the time with our patients. That it just comes back to that's what drives our interest in the lab. So we can have this nice, push forward and also feedback loop from the lab to the clinic. There's certain advantages to that, but of course there's certain disadvantages because it does, I don't want to say limits us, but it does keep our focus perhaps, artificially narrow.
Sure.
And therefore part of the challenge is how to make sure that we still encourage curiosity, to let our science also speak for itself and pose the next questions, not to force the questions on ourselves. So that's a balance that we have in my lab that I'm trying to make sure we carefully tiptoe through to allow both arenas to flourish, but also to allow both to really speak to each other, but not hinder each other as well.
Cool. Yeah. So on the research side, we have a couple questions going over sort of the general fields of cancer treatment that you're involved in. So I guess we can start with immunotherapy. What are you guys involved with in the immunotherapy research avenue? And also, like, where do you see this field going in a few years? Because it's definitely sort of more modern technology that's super exciting for cancer treatment.
Yeah, absolutely, Calder. So that's something I think pretty much most, if not all, of the cancer field is, of course, aware of. And then also understanding how can we harness it? How can we further improve our patients' outcomes by understanding and applying immunotherapy more effectively and designing newer approaches. So first and foremost, it's for sure here to stay. It's now considered essentially a fifth pillar of cancer treatment. Started with surgery, radiation, chemotherapy, targeted therapy, now immunotherapy. So now sort of the five pillars of cancer therapy. And they're all very important. They all have their role, and also various combinations of these five pillars have their role as well. Now, what's quite interesting about immunotherapy is that it's one of the first pillars that was intentionally designed not to directly go after the cancer directly, right? So the first four started, or how it was first developed, was to target the cancer either anatomically or molecularly directly, right? Whereas immunotherapy, the target is not the cancer itself, first and foremost. It's actually on the immune system, or some immune modulation on the cancer that does affect the host immune system. So that's quite fascinating. And that's why I think it's been so beneficial, is that it's just tackling cancer from this whole other approach. It's not directly modulating cancer, though it does, inhibit some of the checkpoints in there or other approaches. Sure. But really, the first direct action is on the host itself. So I think that's why it complements very nicely with the other four pillars, that you're attacking the problem from this other angle. It just gives you more shots on goal. It gives you more, increases your, the effectiveness by having this whole other complementary approach. Now, back to the question of how we're looking into that, is what we've now gained a greater appreciation for, and this is from colleagues and many others people around the world. For example, Shane Harding has shown that with radiation, you can actually engender this systemic innate immune activation, a pro-inflammatory effect. So meaning showing that even local therapies have a systemic effect on the host as well. So how does that interplay in with the immunotherapy pillar? That's something we're also fascinated about. And for us, in the context of lung cancer, with the radiosensitizers, things that enhance radiation effects, do we see those similar synergies with further enhancing a potential systemic response that might improve immunotherapy efficacy? So that's something we're fascinated about, and we have some ongoing work and data in my laboratory to try to figure out what's the best combination, and also to try to test these both preclinically. Of course, testing immune system in mouse models is not easy to do. And also, how do we have relevant models for human cancer, right? We're testing a mouse immune system in a mouse, not really a human immune system. So we're also working with colleagues in the immunology program here at Princess Margaret to try to put a human immune system inside a mouse to be able to test human immunotherapeutics, right? Not mouse immunotherapeutics. So that's another area that we are interested in developing and are active in now.
Cool. So on the radiation therapy side, I mean, I'm more of a physics-minded person, so I'm interested in like the radiation and the technology behind that. So what are some of the approaches you see as going to be really important for radiation going forward? A couple I would think of would be proton therapy for sort of more targeted approaches, or intensity modulated radiation therapy to reduce the dose. Is there any other those or any others that you're interested in going forward?
Awesome. I love talking to my medical physicists and my medical physicist colleagues. So, Carl, they're fantastic. So glad that you're here. You're in a fantastic program with just really true luminaries, and there's so much been done from the medical physics side here in Toronto and here at Princess Margaret, here in MBP. So for radiation oncology specifically, I think from the physical developments inside, I agree with you. I think proton therapy will be important because there are just certain physical properties that you know of quite well, or will learn in greater depth than even I know now, that are helpful in certain clinical indications. And I think we're still scratching the surface of how best to apply that. Now, with of course the challenge, if you remove cost, and the amount of equipment to install a cyclotron and all of that, if costs weren't an issue, I think we would have it, in many, many places at the moment, because there are, certain just dosimetric and physical advantages that a proton beam offers, and that's very different than a photon beam approach. So I think that's one area, and I think it'll be quite exciting to see Ontario, the government, Ontario Health, had just done their health technologies assessment on the need for proton therapy here in Ontario. So, still a multi-year process. Is just one first step, but it's already at least publicly stating the need, and you can put your public comments out. So Calder, you and your students and colleagues and anyone else, feel free to publicly comment on your thoughts on the needs of proton beam facility here in Ontario. So that could be coming in the intermediate-ish future. We'll see.
Cool.
And then the other developments in the physical space, or in within medical physics, I think so. There's this newer approach known as FLASH. What that is this ultra high dose rate delivery. We're still trying to understand. We as a field, are still trying to understand what the benefits will be. There's some studies showing that perhaps it has more normal tissue sparing. And really with that, the challenge of any cancer treatment, whether it's surgery, targeted therapy, immunotherapy, radiation, is this concept known as a therapeutic ratio.
Sure.
We want to kill off the cancer, but not kill off the host, not kill off the patient, right? Right? Because, my one joke is for us in the lab, I can kill off any cancer in the lab. Bleach, pour on cancer cells, and I'll get the job done. Don't worry, right? But that's very different. We can't just give bleach to people or things that would kill off the host, even though that would quote unquote kill off the cancer. So that's not the way to go. So this therapeutic ratio, targeting the disease specifically, but minimizing side effects to the patient, to the host. So that's perhaps where FLASH might take us. It's an area of active investigation for many, many groups around the world. So we'll see where that pans out, but I think that's one other area to keep an eye on. And then the other part, Calder, that might also be interest to you and your student colleagues in this realm is, what's the role of further accurate tumor targeting, not just from an imaging perspective, but perhaps from a molecular or functional perspective? Can we look at imaging biomarkers that tells us where to modulate the dose? What if there's a more hypoxic region that should get more radiation dose, for example? Or for how can we also further leverage digital technology, quote unquote, artificial intelligence to help us do our work more effectively, that is less manual in nature, and perhaps can even optimize a plan or come up with the best way to target or deliver the radiation in a way that we couldn't think of as a mere human mortal. But that's still a long ways off before, computer technology can really help us do our work. But I think that's still an area that is exciting to develop further. We have proton, flash, AI. I would say those are, and then functional imaging will be
Sure.
very exciting areas to look out for. And actually, the fifth area that I would implore us to really think about is within radiation oncology, even for my medical physics colleagues, is also not to neglect, but also to actually think, to have some appreciation for perhaps, but maybe also some awareness of biology and how does that interact with our physical treatment, right? With our ionizing radiation. I think that will be quite important because if we can manipulate and harness our understanding of cancer biology, I think it will help us deliver and utilize our modality and all those other forms I just mentioned, proton, flash, right? AI. I think it will actually further improve how we treat our patients, because no one patient's cancer is alike to the next patient. Every patient is unique to themselves, and even the biology of their cancer is unique to themselves. So that's another area I would highlight for us.
Cool. Yeah, I mean, you segued yourself really nicely into the question of DNA repair, because that's all about sort of what's the combination
Perfect.
between radiation therapy, cell biology, how the two interact. So yeah, tell us a bit about the DNA repair research you've been interested in over the years.
Absolutely. So, our cells, they suffer, endogenous DNA damage day in and day out. There's just metabolites, oxidative stress, and our body, and our cells have really developed these very intricate ways to keep our DNA intact from these daily assaults on our genome. And then also what's been this whole other layer to it is, with the advent of genome sequencing, that has led us to unravel that cancer at one fundamental level, it's truly a genetic disease, an altered cancer genome. It's acquisition of different mutations and altered proteins that are encoded from mutated DNA, right? So that's what the cancer harnesses, is this power sort of like microevolution within the human body. A lot of those cells are killed off. The body has mechanisms to clear those out. But quote unquote, the lucky few, or unlucky for the host, acquire certain changes within its DNA that allow it to become malignant. So that's where DNA repair is also quite fascinating. Within cancer, it's either hindered it or harnessed it to become part of the cancer process to confer a survival advantage. And I think one of the first, proof of cancer being a genetic disease is sort of how the whole DNA repair field is also very closely linked to, is these breast cancer susceptibility genes, BRCA1 and BRCA2, that we hear a lot about. And they're very important for genome stability and double strand break repair of broken DNA. And with loss of that, for patients that are born with or carriers or one copy of BRCA1 or 2, or mutated, altered, or non-functional, they have a really, really high chance of developing breast or ovarian cancer, and also other types of cancers at a lower frequency, but still showing how closely linked DNA repair, genome stability is with preventing cancer from developing. And now the third layer beyond that is actually how do we, knowing this, can we also exploit that by understanding biology? So that's where, for example, in BRCA mutated ovarian and breast cancer, PARP inhibition came about. Was this synthetic lethal interaction, meaning if you target the single strand break repair pathway that's PARP mediated in cancer cells that have or deficient in BRCA pathways. They are more sensitive to the treatment. So, I think just all these layers are quite fascinating. So how are we harnessing that directly in my laboratory? We are looking into different resistance mechanisms where it seems to upregulate double-strand break repair, specifically through this homologous recombination pathway. And just diving into both the mechanism and the observations that we've made, we're trying to develop therapies against what we see that's driving this. We found this interesting SCIF E3 ubiquitin ligase that mediates homologous recombination through targeting different proteins that are involved in the pathway. So we're trying to work that up and really dive into the mechanism of that and also translate that into a potential way to either prevent that resistance mechanism from occurring or even targeting it, perhaps through synthetic lethal routes. So that's how it informs what we do, and it helps us give us a lens to look at cancer biology and also cancer treatment, and gives us a focus to our laboratory and how we want to apply this out to the clinical problem that we see.
Cool. So switching gears a little bit into the more sort of lab environment side that some of the people listening to the podcast might be interested in. So what sort of qualities would you look for a grad student to enter into your lab and really succeed there?
Great question, Calder. So to succeed in my lab, partly is quite interesting. I am a junior PI, so we should probably ask some people wiser than I that have done this longer. I'm still developing, I think, how my mentorship style is and how best to coach and mentor students. But I think one thing I've also recognized is to, in a sense, personalize it for each trainee, because everyone's different, right? I'm different, you're different. Every student that comes is different. So first and foremost, I think it's to recognize that there won't be a very cookie cutter way to do things, and even for the mentorship side, and even of how that success develops. But what I can say is that there appears to be certain ingredients that will help increase one's chance for being successful. I think one thing is to have a passion in the research that you're about to jump into. It's not always love at first sight sometimes. There will be times when your passion towards your project and your science, I think, would make sense, would wane. Just like for those in romantic relationships or married, you don't always have these warm, fuzzy feelings towards your partner, or even your hobbies that you enjoy. I don't know, let's say you really enjoy a certain sport. You don't always love it every day. But I think it's also to how do you keep yourself motivated even during those times where you don't love your research topic, you don't love your project. But I think it's how you sort of get through that is to have the big picture in mind and have the broader view. And it's something I sort of tell my students and those in my lab is that, I think beyond your focused research project is to understand that in academia, in a scientific laboratory, your task, your mission is actually to add to human knowledge that has never been seen before. You are literally explorers at the edge of the known universe of human knowledge, and you're about to take a step into the unknown. So that, at least for me, is what keeps me motivated. It's not so necessarily about, oh, this project didn't work out this way, or, oh, man, I'm beating my head against something that needs to be optimized. And it's very hard work, to be an explorer of the unknown, right? You're literally taking steps into the void. Take pride in that. Have that really gird how you approach things, because if it's about your very narrow focus in and of itself and you're missing the big picture, I think it's easy to just feel beaten down a little bit, because it's hard work to step out into the void. It is. So that goes into that next part is first, have that big picture in mind. And the second, then now into the day-to-day, into your hard work, know that it takes hard work to be that explorer at the edge of the universe of human knowledge. That's what you're doing now, Calder, as you are designing your experiments. That's what every trainee is doing. That's what every lab is doing. We are at the edge of the universe here. The other piece of advice I give folks is to measure twice and cut once. A well-designed experiment is worth its weight in gold. It's not simply hard work. It's not simply just how much you pipette one day or how much you code in the lab one day. It's actually how well did you design your experiment to answer a question. And even if it doesn't pan out the way you expect it to, either from a technical side or from what the data are in the end, you should have still learned something from each approach or each attempt. And even if it's just missing a basic step that just caused the result to be uninterpretable, you would have learned something. So I think that drive, that perseverance, keeping the big picture in mind, and leaning on other students, vent to each other, but also inspire each other, and just share knowledge and just share excitement about what you each are doing, even on the days where you just feel like you just really don't like what you're doing. But drawing in that well to persevere and keeping the big picture in mind, I think helped me, is what I would say. And it might help some of your listeners as well.
Cool. I think I'll just do one more quick lifestyle question before we get into the fun stuff. So imagine, let's say, I don't know, a year ago, Calder wanted to do a rotation in your lab. What sort of project do you think I could be able to accomplish in a 5-week time frame?
Yeah, great question. What I will say, 5 weeks is really short for any lab project,
right.
but
Absolutely.
But it's more than 1 week. It's more than 3 days.
True.
In my lab, it would be to get a flavor for what the potential thesis project could look like. We have a pretty broad portfolio focus within lung cancer, running from, even from liquid biopsy and some computational analyses of the resulting sequencing data, to in vivo animal models. So that's probably not a good rotation type of approach. You could literally spend the whole month learning how to work with mice and orienting to that. That wouldn't be, unless someone had a lot of animal experience prior to coming in, that wouldn't be a type of project or part of the project that I would put a student on. But probably also in the middle, I would say in the wet laboratory, is some of the basic in vitro work that we're doing. Sort of novel therapeutic development, drug response type of data. We have this new exciting epigenetic chemical probe. We want to generate some more replicates to this. So work with our fancy robotic drug printer that we have and our flow cell sorter and generate that data. And within your month, you can actually have done probably a couple or a few of those and sort of see some tangible results from your effort. So that's one type of potential rotation project. Other rotation project will be to help generate and work with some functional genomic technology. So CRISPR-Cas9 to knock out different things, or use shRNA to knock down different things, or overexpress. So just basic molecular biology that would help with any future wet lab type of project. So those will run the gamut. And I think it would depend on, I don't want to say month to month, but every quarter, there's different activities within these types of projects that a rotation student can become involved in.
Cool. Sign me up. So, a couple more off-topic questions now. So what do you do in your spare time unrelated to your clinic or science work?
Now with the pandemic, it's a very weird, time, right? There's only so much we can actually do. Yeah, I mean, pre-pandemic, I would say I quite enjoyed just going to the movie theater, just checking out the latest release. That was a nice little fun thing I used to like to do. Haven't been able to do it all this year. For now, from my home life, I'm quite busy with 2 young children. They are not yet really fully in school at all, so they keep up a lot of my out of work time occupied. So sadly, not a lot of time for my own personal hobbies, but I try to make do.
Cool. So, an alternate universe where you didn't become a clinician scientist, what sort of jobs do you think you would have ended up in?
Yeah, good question. I like this question. I think there's a range of some more like aspirational to some more pragmatic. I could probably answer a little bit of both. Maybe you get a sense of it when I was talking about human exploration, edge of the known universe. As a young kid, always was fascinated with outer space and whatnot.
Mhm.
So growing up, I thought, oh, maybe an astronaut would be sort of cool. Uh, my dreams were dashed though because I realized quite early on, even when I was younger, I actually get some sense of motion sickness in moving
Oh yeah,
vehicles. So I was
that's
like, ooh,
hard.
I don't think that will quite work out for this as a career path. I wouldn't make a great astronaut.
I don't
With
think I
that,
would either. None that reason.
I'll fall over at the controls and
Mhm.
pass out. Um, but that and also it's a very selective career route, but one that would have been fascinating nonetheless. Probably on a more practical side, maybe it would have been in the business world. I don't know. That was my undergraduate work. It was a very practical type of major, and might have stuck around like some of my classmates did in the business world, and I don't know, working on Wall Street and shorting GameStop stock or something. I don't know. But that might have been like the pragmatic thing. But who knows where life would have taken me? It's sort of quite interesting to know that we live life only one way. It's moving forward one way, and that's and I'm here now, and I, in a sense, uh, it's very cliche to say, but I have no regrets, so to speak. Uh, Quite enjoy the process, and even if I don't become as enamored with my current career path, I always remember sort of have the big picture in mind. It's not simply about the day to day. So hopefully, it'll get me through whatever future challenges arise. Overcome some to get here, overcome some on a daily basis too, of course. But that's something I bear in mind. But anyway, to your question, astronaut or some sort of practical business job, perhaps.
Cool. Then what's a sort of special skill or talent that you have that most people, say outside your lab or outside your family, wouldn't know about?
Yeah, for outside my lab, I had a lot of how is this like amateur training in music.
Mhm.
Um, you know, Nothing nowhere near professional at all. But I was very fortunate to have parents that would just help expose me to different experiences in the arts, in music, in sports, in science. Actually, the one story is I was sent to the Cold Spring Harbor Laboratories, a very famous lab in Long Island, New York, in the eighth grade to go to this like DNA gel camp or like DNA camp. So I actually made my own agarose gel in eighth grade, and so like I think I really credit my parents for just this diverse upbringing, and with that came a lot of uh, maybe not so random, but a lot of just musical things. So growing up, played the piano, trombone, violin, like sang in the school choir. I picked up guitar on my own in college, then like acoustic guitar, and spun out a little bit into electric guitar just to play around with. With all that background, like you can sort of mess around on a drum kit, so I can play the drums a little bit as well. Um, so I wouldn't say like I'm a multi instrumentalist, but you with piano, guitar, you can sort of make your way around. And actually, with the brass instruments, I could probably pick up some. My brother played trumpet, so I could like noodle around on the trumpet. So that's sort of cool. I know my way around quite a few different instruments, and sang in the choir or something. So I have some appreciation for um, how hard it is to be a professional musician. There's no way,
Yeah,
I'm not even close to
absolutely.
anything like that. So very much amateur in my experiences,
Mhm.
but I can make my way around a few different instruments.
Yeah, I play well I play, I guess it past tense, played saxophone a bit. And a
Nice.
bit
I
of
love that
piano,
instrument.
but yeah, definitely nowhere near good. My favorite was actually doing pit orchestras for musicals. That
Yes,
was a
I
lot
did
of
that
fun.
once. That was a lot of fun. Yes, that's right. So was it saxophone for yourself? In the
Mainly,
pit
yeah.
orchestra?
Mhm.
Nice, yeah. That was me with I think it was trombone. Yeah, trombone.
Yeah.
Yeah, pit orchestra. Yeah, for the school play. It was fun. You get that? What was it? I think it was for Fiddler on the Roof. That was the play I played in.
Nice.
I guess what you enjoy about it, Calder, being in the pit?
What do I enjoy about it? So first off, you're not being looked at like you would be on the stage. That's cool.
Right,
So
yeah, we're not on the stage. Yeah,
there's a
those
lot more
guys up
freedom
there.
in terms of just messing around and whatever. But also it's fun because especially as you practice more and get more in tune with the show, then it just becomes a lot more fun of like you learn all the songs, you know which ones are your favorites. You'd really enjoy playing those. Yeah.
Absolutely, yeah. I have fond memories of that. It's also you get to know the play really well,
Oh,
and also
absolutely.
when
Yeah.
they mess up on stage, and also when we mess up too in the,
Right.
in
Yeah.
the pit.
Fun.
Great, awesome.
Cool. Yeah, I guess, do you have any questions for me? Since I've been asking you questions over the past half hour or so.
I'm just welcome to the department.
Oh, thank you.
For yourself, you were born and raised in California or elsewhere?
Yeah, so I was born and grew up in San Diego, California, and then went
Nice, I
to
love San
undergrad
Diego.
at yeah, it's a great place. Went to undergrad at U Chicago though, so definitely a little
Oh,
bit
went to very
chillier
cold.
there.
Yeah,
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 well,
very different. Glad you're here, Calder. And then, you've done rotating? Is that right?
due to the pandemic, we had sort of an extended
Direct.
interview process where
Ah,
it's
yeah.
like over the summer we were given a list of faculty who were recruiting, and so we could send emails back and forth or arrange virtual meetings. So I kind of did that, and then found my supervisor, Dr. Graham Wright, that way.
Oh, Graham, right? Yes, I know him. He's I think he used to either mentor or work very closely with Jay Detsky, one of my colleagues now up at Odette Cancer Center.
Hmm,
He did a
cool.
PhD in like MRI cardiac imaging. Yeah,
That's what
pretty
I'm studying. Yep, cardiac
perfect.
MRI.
Okay, I think my buddy Jay Detsky was with Graham in his lab or his collaborator, something.
Sure. Yeah,
Cool.
there's a wide net.
Actually, speaking of cardiac imaging, there's a for my clinical practice, just to let you know,
Mhm.
something we're um, trying to develop has been this technique to actually um, counteract cardiac arrhythmias
Yeah.
or to ablate arrhythmias. Normally, you would do it by the cardiologist would snake a catheter up into the heart
Yep.
chambers and sort of zap away, or use RFA or microwave energy, what have you, or even cryo
Mhm.
to ablate some of the arrhythmogenic foci in the heart. Now, for those that can't tolerate that procedure, is too high risk. There's been this very new approach of using radiation
Oh yeah.
to actually ablate that. So maybe offline, I'd love to like pick your brain, or maybe I'll
Sure.
chat
Because I
up
know,
Graham
what is
again at
it,
some point.
This is more like Dr. Angus Lau's research area. I think he does a lot of MR guided radiation therapy stuff.
Cool. Yeah, yeah. So anyway,
Yeah,
yeah,
definitely should talk
maybe we'll run
offline about
into one of
that.
these days about it, or yeah, or
Fun
chat some
stuff.
more about it. Yeah, because we have MR Linac at the Princess Margaret. I know Odette has one too,
Yeah.
and we're also trying to figure out what to do. But then again, a lot of these patients have pacemakers and stuff, so
Right. Yeah.
it's a
Gets a
bit
little tricky
tricky.
there.
Yeah, yeah, exactly. Well, Calder, this was great to be interviewed by you and to chat with you.
Awesome. Yeah. Thanks
Um,
for coming on the podcast.
Absolutely. So hopefully it captured what you needed, and that this is useful for your students. And also, I know there's probably viewers from outside MBP, though I'm sure most are within for now, until you guys go viral or something, where this just like
Actually, I think
blows up the internet.
a lot of our target audience is sort of incoming students because
Mm,
it really helps give a sense of who are some of the faculty, what is their research area, what do they like outside the lab. Yeah.
great. Okay. No, so that's great. Thanks for doing this on behalf of prospective students and your future colleagues.
Yeah, no
So that's
worries.
wonderful.
Hope to talk to you again sometime soon.
Absolutely.
Cool.
Our
See ya.
call. This was fantastic.
Great.
Bye now.
Later.